Hello, welcome to lesson 6.2. This lesson will focus on Amoebae as one of the protozoa groups that we will learn about. We'll first of all talk about the survival mechanism and the reproduction and dispersal, which is via cysts and spores. Just to put it into context that these organisms actually have to have survival mechanisms in soil because they are normally aquatic organisms and they need moisture and water in order to move and reproduce and have a lifestyle. We'll talk about testate amoebae as these form shells. So that's another way of actually protecting themselves from drying out and just being in the soil environment. And lastly, we'll talk about the motility of these organisms and how amoebae move via these pseudopodia, which are these foot-like extensions that they form in order to move. Okay, so let's get into lesson 6.2. One of the most important things to remember about all the different types of protists is that they're actually aquatic organisms. So all of them have come from water. And so this means that they are very reliant on having moisture in the soil in order for them to survive. So all of these organisms are actually water-based or aquatic organisms. And there are various ways in which they need to be able to survive when conditions get really tough for them to live and water is scarce or non-existent. So first of all, let's look at the life stages of soil protists. In this image, we can see the different types of heterotrophic protists. So they are the ones that actually consume other organisms, particularly bacteria, releasing their nutrients for plant availability. And as I've introduced in lesson 6.1, these include flagellates, amoebae, and ciliates. The interesting thing is that amoebae and flagellates can often, in some species, be interchangeable. So the organism can move using either a flagellum and becoming a flagellate versus an amoeba. So this is very interesting and strange, but the shape-shifting amoeba can even further shape-shift into a flagellate. And that must be a fascinating process of how that occurs. But different life stages of different species may involve these two different types of locomotion and being. Ciliates are a little bit different. They have cilia to propel them. They move really fast. The cilia are little hair-like projections on their bodies. And the amoebae move through these pseudopodia, as I've described in the last lesson. And the flagellates have these whipping flagella that allow for their movement. Ciliates are one of the largest groups. They can get quite large, actually. And amoebae can be quite large as well. Often the flagellates are much smaller. So because the soil matrix is actually very prone to wetting and drying, ciliates prefer to always have wet conditions. And so there are more ciliates in soils that are constantly wet, whereas the other organisms are often seen in quite even drier soils as well. But it's got to do more with the oxygen availability. These guys, the ciliates, can actually handle reduced oxygen conditions in the soil, and so they will proliferate, whereas these guys will move into a cyst stage if the oxygen conditions drop below their requirements. So in all, because the soil can be quite dry or even anaerobic and food can be scarce at times, these organisms are really good at forming cysts. And cysts are these dormant stages of the organism where all or most of their metabolic functions are closed off or shut down, and the organisms remain for some time and sometimes long periods of time in a cyst form. And there are different types of cysts that these organisms can produce. Amoebae, some of these amoebae or flagellate organisms, form slime molds, what we call slime molds. These slime mold structures that we call plasmodia or plasmodium. So plasmodium is when these guys get together when the conditions are right for them to actually want to sporulate, to reproduce themselves. So these organisms that just dwell in the soil as singles can now get together and form this giant cell, the plasmodium, that has multiple nuclei. So these cells just grow through these additional nuclei being formed, these giant cells that are visible to the naked eye develop. And then at some stage, when they finish feeding on fungi and other types of materials, wood, whatever they like to eat, once the food source is gone, they will produce the sporangium. And the sporangium is what contains these spores, and the spores are ejected out of the sporangium and the life cycle begins again. Interestingly, individually, the flagellates and amoebae will feed on bacteria in the soil or other smaller protozoa and algae, but as a plasmodium, they consume fungi. There are also different types of photosynthetic protists that are unicellular as well, and they also have a cyst stage in their life cycle. But we'll concentrate more on the heterotrophic predatory types of protozoa, and we'll touch upon some of the photosynthetic protists as well, but not in as much detail. So cysts, survival and dispersal. Cysts are basically formed when food or vitamin deficiency occurs, when there's crowding, where there are bacterial and protozoan excretion products that might cause cues for these organisms to start forming cysts. They're basically compromised in some way, and the conditions within which they find themselves are no longer conducive for their optimal function. And this might include desiccation, so drying, salinity, extreme pH, reduced oxygen, and sometimes even under optimum conditions, when food is in excess, the organisms might actually encyst as well. So it just depends on the species and what is going on within the ecosystem that they're in, and what their environmental cues are for them to decide, okay, it's time to exit. I want to go into a metabolic shutdown, produce a cyst, and stay in that form for potentially many years. So some flagellates and amoebae have been revived from their cyst form after being in dry soil for 50 years. Some amoebae cysts have been revived from late Pleistocene permafrost, and that means they have been locked up in a cyst form for 30 to 60,000 years. So these are very hardy structures that are made by these organisms in order for their survival to be achieved under extreme conditions and long periods of time. Extreme temperatures, where the soil can get to below 11 degrees Celsius for a year, can also produce revival of these cysts. Wet heat of 50 degrees and dry heat of up to 120 degrees Celsius is also survivable in a cyst form. Outdoor air may contain 1 amoeba cyst per 9.1 meters cubed, and several soil amoebae are actually airborne and have been studied in the dust storms in Nigeria. For them to be transported, other than being airborne, some of them are consumed by soil animals, and because they pass through their gut undigested, they end up moving to different places. So this is one way that soil protozoa can actually move, by being eaten and pooed out in their cyst form. So on the right in the diagram, we can see the feeding cycle and self-division of the vampirellid amoebae. And as the name suggests, these guys are likened to vampires because some of them can actually latch onto a cell and suck out its contents. And this is basically just to show how they can go into this active searching phase where they move and reproduce and feed, and when conditions are unfavorable, they go into the inactive phase, which are different types of cysts. A true resting cyst, where all the metabolism has been shut down, is the spore. So a lot of them will produce and survive via the spore stage. Here you can see a vampirellid amoeba in its active searching phase, which is called trophozoite. And then when prey is available, they will feed, and then they will form a digestion phase where all their pseudopodia are intact and they're digesting the organism, usually a bacterium they just fed on or another protozoan. And then they will go through a reproductive stage where there is the duplication of the genetic material, karyokinesis, which is this K phase, which occurs normally in the late digestive stage. So reproduction occurs and eutrophozoites then hatch. So this process is called plasmotomy, and this allows for asexual reproduction in which a multinucleate protozoan cell divides into 2 or more multinucleate daughter cells and basically replicates itself, clones itself. And so these cells are then released and become the active feeding stage again. And they will go off and search for food again, and the cycle continues. And if the prey runs out, they will form the inactive phase as one of the cysts pictured here. So very fascinating creatures, a very interesting life cycle these organisms have. Very, very interesting. Besides cysts, protists have also other ways of ensuring their survival. For example, testate amoebae are amoebae that actually create or secrete shells around them, and they hide in those shells. These guys are non-pathogenic. They live in all kinds of environments, and there are between 2,000 and 3,000 described species. They range in size from 10 to 800 micrometers. So that's 1/10 of a millimeter. That's how tiny they are. You obviously need a microscope to see them. And they constitute some of the oldest eukaryotic fossils. And it's been estimated that they have been around since 1 billion to 730 million years ago. And they're basically exactly the same now as they were back then. They resemble modern species. So those fossils are basically identical to the kind of testate amoebae we have in the environment now. They preserve well because of the shell, and that means that they can be used as environmental indicators because they can show us what was happening in the environment at the time when testate amoebae shells were deposited and then transformed into fossils. The saprotrophic amoebae that feed on organic matter might be the primary decomposers when bacteria are less available or unavailable because of some particular environmental scenario. And so these guys are really important as saprotrophs, as nutrient cyclers, not just as predators, but also as primary decomposers at times. There is greater relative abundance of these guys compared with flagellates or ciliates when there is less moisture in the soil. Because they have the shells, they're able to avoid being dried out, they can avoid desiccation, and they can hide. In the image on the right, we can see the complex ornamentation that this particular testate amoeba has created using all kinds of various materials, various diatom shells it looks like, and other things in order to create its shell, in order to make its little home. So this is the kind of tests that some of these amoebae create. And some of them have ornamentation such as little horns and things. And the amoeba is actually lives inside of this test. Okay, so let's look at now the ways in which these organisms, these testate amoebae, actually form these shells. So testate amoebae produce their own tests, so their own shells. And when they secrete their own shells, or the units to make the shells, these shells are called autogenous, self-made. And these are often proteinaceous, calcareous, and some of them might be made up of silica. Some of the amoebae, such as Arcella, are examples of autogenous tests, which hide within a very round, globular, hollow shell. They are termed areolate. So that means just round. And these rounded shells are proteinaceous, so they're made out of protein material. Other testate amoebae create xenogenous shells, so that means they actually glue organic or mineral particles, such as sand grains and diatom shells, to themselves. And these shells are also called agglutinate tests because they are glued on. And the glue, by the way, is produced by the testate amoeba. There is enormous arrangement and diversity of the actual structure and how it's composed. And the patterns vary from like fish scales, like you can see here on the right, and little squares or little circles. They're just incredibly beautifully ornamented, as you'll see in a minute when we go through some of the images of different testate amoebae. Some of the amoebae actually prey on other amoebae that already have a shell, and they will steal that shell, or will recycle the materials made from an amoeba that is no longer in the shell. But some of them are essentially like crabs, where they'll hunt the animal from within the shell and just reuse their shell. What's interesting is that those amoebae that have flatter shells display more activity in thinner water films. And this is, of course, because they are adapted to a drier environment, and their shells allow them to survive in less water as a result. So they can actually still function as the water film gets thinner. Because remember, these organisms are aquatic and they need water in order to move. So this just demonstrates how important it is that the structure of the organism reflects what type of environment it lives in. So the organisms that have these flatter shells are able to withstand drier conditions as a result of having that flatter shell, and therefore feed for longer periods of time and be able to nutrient cycle to the plant. It's just very important to always relate the structure of the organism to its adaptation to a particular environment that it dwells in and functions in. So these are just images to show you the various beautiful formations that the different testate amoebae can create, and how intricate it is to glue all these particles onto itself. These shapes are diatoms, so they're also protists, photosynthetic protists. And obviously when they die, their shells are left behind, and these testate amoebae can pick them up and adorn their little homes with these particles, these exoskeletons of these diatoms. This is an amoeba that has a beautiful square arrangement of its particles on its shell. It just boggles the mind that a tiny creature, just one cell of this strange little shape-shifting organism, can actually form this incredibly elaborate and perfectly arranged shell. So these are just different microscopy techniques that are used to show the organisms. Here's one of the amoebae. Looks like a snail, doesn't it? It comes out with the pseudopodia. This image depicts a process called binary fission, whereby the individual testate amoeba is able to clone itself by producing a daughter cell, and it basically goes through a cellular replication to make an exact copy of itself. A fascinating process. Like bacteria and other protists, that is one of the magic tricks that an amoeba can do. It can just basically clone itself. So testate amoebae live in their shells, but they're able to extend their pseudopodia, which are foot-like projections, to allow for their movement. So normally the organism rests inside the shell or the test, which is also called theca, and then when it wants to move, it will extend these pseudopodia from its shell, from the aperture, which is the entrance to the shell. The organism is attached to the test by epipodium, so these attachments, and the pseudopodia have various shapes. And here are the different types of pseudopodia that different species of amoebae are characterized by. Lobos or lobopodia are lobe-like with broad blunt ends. You can also think of them as finger-shaped tubular pseudopodia that are featured here in this photo. There are phyllos-shaped pseudopodia, which are called philopodia, and they are fine, thread-like, and tapering. So this is an example, an image of philopodia here, kind of networked, very thin. And then there's also axopodia, and these are hard and stiff, forming axial filaments. There are also reticulopodia, which form networks, and I think it's the same here as the granular reticulose pseudopodia. So they all have various ways of extension and movement and how they behave, these pseudopodia. Like, for example, the reticulopodia actually branch and form these extensive networks that you can see here. So this is how a testate amoeba moves. So if you want to learn more about these extremely interesting creatures, there is a fantastic site called arcella.nl, and basically there is a man who studies amoebae, all kinds of amoebae. He's from the Netherlands, and he's basically got this website, and you can learn a lot from that website. It's a very, very well done representation. He's got a lot of microscopic images of these guys and videos, and it's very interesting, so I highly recommend checking out his website. So just to describe this amoeboid motility, or the movement of amoebae via the pseudopodia, which occurs at 0.2 to 3 micrometers per second, depending on the species of the organism. To show you this, I'm going to play you a quick video of a naked amoeba, and this is an example of a naked amoeba without a shell, just to show you how these organisms actually move, how the pseudopodia function, and how cool it actually looks. It's a video of an amoeba that I took some months ago, and then I sort of sped it up just to show that incredible movement of how the contents gets transferred from the cell into these pseudopodia extensions. That big animal that came through was a rotifer. That is super cool. Okay, so this is the end of lesson 6.2, and we are going to learn about flagellates and ciliates in lesson 6.3. So we'll look at those two groups of organisms, in addition to also microalgae as a very important component of soil protists and the soil ecosystem. Okay, so you'll be hearing from me in lesson 6.3. Bye for now.